Well, hello. Now, we're going to start week one, covering some of the concepts of disease emergence and reemergence. This is for the emerging and infectious disease course for Eastern Florida State College. And I am your professor, Dr. Lawrence Roberge. Now, first thing is first. We got to make sure we have all the ducks lined up for all the students. Students must have the required textbooks. Now, these are going to be in ebook format. Students will also have a variety of research papers, articles, and videos in each week's module. Students are required to read and review these attachments that are going to be in the specific modules. Therefore, students must plan ahead for each and every week. You will have the opportunity to have a review quiz, and after it's been graded, I encourage students to review not merely the correct answers. Yeah, you got them correct. Let's say you get 80%. That's 80% you got correct. But what about the 20%? Therefore, I encourage you to take the time, review the incorrect answers to learn what you got incorrect to avoid the same errors in the subsequent exams. You are expected to review the readings for each week as well as the lecture. The PowerPoints for each lecture will be available in the weekly module, in the module for that week. Plan ahead on the discussion assignment and select the topic early in the week. When you have a situation where I tell you you've got to choose a disease, for example, for that particular week, what you want to do, so no two students can have the same topic, is stake your claim early. And all you have to do is just put in the one or two words that are the name of the disease, and that way that is sequestered, or you've locked that in for yourself, and then you're going to basically write up your discussion. You will also have a commentary post. Now, how to do the original and how to do the commentary posts, all of this is due by the end of the particular week. And I also give you basically instructions on what is considered important in the original and in the commentary posts. One of the key things is always cite where you got the information. Now, we're going to do this in the proper format, and the format tools will be present in the announcements section. Okay? No late assignments are to be accepted. The weekly quizzes must be completed by each Sunday at 11:59 p.m. Eastern Time of that week. For exams, those will be available for a selective 24-hour period only. Also, review the announcements section for extra tools to help you during the course. And as I mentioned earlier, you're going to have a variety of videos, etc. I try not to get the videos greater than 10 minutes. Some of them are nothing much more than 2 minutes, 5 minutes, 8 minutes, etc. But you're expected to view all of them. And you're also expected to read the papers because they're going to be very relevant as you take the time and dive deeper into emerging and infectious diseases. Now, there's terminology that we have to go through, and I'm going to mention some of the terminology and some of the issues and concepts of epidemiology in this section. When I talk about infectious diseases, these are illnesses caused by germs. Now, germs can be microbial organisms, bacteria, viruses, fungi. They enter the body, multiply, and can cause an infection. Now, some infectious diseases are very contagious, or we would call it communicable, meaning that it can be passed from one person to another. Now, this is very important because as we go onward, we're going to talk about R naught. In other words, the capability of the disease to spread to other susceptible individuals. But let's just step back a second, and you're going to see a bunch of papers that I selected for you, and they are talking about the evidence going back to, in some cases, prehistoric times about how some diseases have been found. But the people at that time, roughly until about 1860s, many people believed that disease was spread by bad air. That's why you're going to have terms like miasma or malaria, which actually is derived from the term bad air. Now, we started to move away from this when we had Anton van Leeuwenhoek, who had first made the first functioning microscope. It was a simple single lens, but he was able to study all sorts of different microbial organisms that you could not see with the naked eye. Okay? And he called some of these animalcules. Now this is the 17th century now. Then we're going to move into basically the 19th century because there was still some confusion. And then we have the work done by Louis Pasteur. Okay? Now, Louis Pasteur disproved the bad air theory completely by his work resulting in the germ theory of disease. The germ theory of disease is basically, it's a science-based explanation that infectious diseases arise from exposure to microorganisms and germs. Now, to jump further onto this, in 1884, Robert Koch developed Koch's postulates, and this is to assist whether a particular organism was the disease pathogen. This is extremely important because if you say to somebody, okay, give me a throat swab or, sputum or blood or whatever, you might find a bunch of different organisms. So which is the organism that is actually making that individual sick? Now, Koch's postulates are the following. Number one, the microorganism must be found in disease, but not in healthy individuals. Number two, the microorganism must be cultured from the diseased individual. Number three, inoculation of a healthy individual from those cultured microorganisms must recapitulate, or in essence, replicate the disease. Now, 4 basically states that the microorganism must be re-isolated from that newly inoculated and diseased individual and matched to the original microorganism. Okay? Now, you might sit there and say, isn't that kind of long and drawn out, etc.? No. You have to understand that when you're dealing with individuals who have been under this influence of bad air or something else that causes it, what you have to do is, by the way, when I say diseased individual, most of the time we take, let's say, a staphylococcus or a virus, we put it into healthy mice or rats, or we might put it into monkeys or rabbits. Depends. But the idea being is this. Let's say you have a diseased animal and you find it only, this microorganism, only in that diseased animal, not in healthy animals. You culture up the microorganism. Then you make that culture and inoculate a healthy test animal with the cultured organism, because that is going to basically reflect the disease. Now, the microorganism must eventually be re-isolated from the inoculated diseased animal or individual and matched to the original microorganism. Now, there's a couple of caveats here you got to understand. We have to use, in some cases, animals. Why? Well, you're not going to get tons of people who will say, okay, you can inject me with some sort of pathogen that has a 50 or 80% fatality rate. Nah, it doesn't work like that. Okay? The other challenge is this. Some microorganisms do not live or proliferate in certain test animals. Some of them basically will be found to culture well in minks or chinchillas or rats or mice or rabbits, etc. And this is where you have to really do your homework. Now, excuse me, the thing is, you don't want to take something as deadly as AIDS and inject it into a normally healthy individual. Now, since cautious work, we've had definitions of diseases with varying nuances. Okay, let me give you what I mean by varying nuances. We're talking still about COVID. And now COVID has mutated a little bit more. Now they're going to call it, not by the letter code, but they're going to be a combination of this and Greek symbols or Greek names. And that's because those changes have convey certain nuances, certain capabilities to infect or proliferate in the respiratory system. Okay? Then you have something like influenza, where influenza undergoes basically mild mutations. Hence, that's why we have to go through the flu vaccine each year, because there's been either subtle mutations or profound mutations that have made our previous vaccines useless. Okay? Now, certain terminology. When I talk about something asymptomatic, A that you put before something means without. So you have no symptoms. You may have individuals who are infected but do not show the clinical symptoms of the disease. This is in contrast to symptomatic individuals. Symptomatic, that term, that's where individuals are infected who exhibit clinical symptoms of the disease. One interesting view is this. If you take syphilis, and in the early part, infected individuals will get a small, it looks like a sore, or not a blister as much, but more of an open sore. Now, that could be on any part of their body. A lot of women were carrying syphilis, and where that sore was inside, either in the uterus or in the vagina, and they were not aware. Whereas with males, it might be on the penis, it might be somewhere else on the body. And that was what we see then. Nowadays, we see things a little bit differently. It's a quick blood test. Okay. Now, let's talk about the symptom. When I talk about symptom, there's sometimes you're going to get symptoms and signs. And a symptom is a physical or mental feature which is regarded as indicating a condition of the disease, particularly such a feature that is apparent to the patient. Rocky Mountain spotted fever. You're going to have all these looks like little basically speckles on the skin, and that's really called petechiae, where small blood vessels are broken, particularly capillaries. Okay, that's Rocky Mountain spotted fever. Now, someone who's a carrier is an individual who is infected and is able to spread the infection to others. It was very common for some individuals during the early years that we had AIDS that an individual would be a carrier. They're infected, but you see the effects of HIV don't really show up for 6 to 9 months. And part of that reason is that, yes, unless you're looking for it, unless you pull a blood test or something else like that, what's happening is the immune system's fighting, and only after the 6-9 month period, the immune system is falling apart, that you start getting all of the opportunistic infections. And what I mean by that are things like Kaposi sarcoma, and you have mental effects, and the individuals may have lung infections, etc. These would be diseases normally that we would brush off if you're healthy. Okay? Now, infectious diseases are caused by microorganisms, as I mentioned to you before, such as viruses, bacteria, fungi, or parasites. Now, another group came on board, and if you ever get to read some of the stuff by Stanley Prushton, he talks about his early work and eventually getting to the point of beating off the skepticism and eventually coming to the end point of getting the Nobel Prize for this brand new, if we want to call it that, it's not really true, but it's an infectious organism that's not alive. It's infectious proteins, and they're called prions. Now, the difference with prions is they have no nucleic acid. So the previous attempts at inactivating the pathogens wouldn't affect prions, okay? And it is passed and it can cross the species barrier. So you can start off with sheep having it. They end up with a disease called scrapie, where they basically have, as they're having this spongiform encephalopathy, meaning that the brain, a lot of the cells are dying inside. So it looks kind of spongy. They will have repetitious behavior, one of them being they will rub themselves furiously against something like maybe a post or something like that, to the point even that they have not only scraped away all of the wool and the skin, they'll actually have an open wound. Now, it used to be that we would take and render sick animals and grind them up and make them into a high protein meal. And this happened more with Europe than with the United States. But to provide that protein meal, which was fed then to cattle, unfortunately, what you have is the prions jumping from scrapie infected sheep to cause what we call generally mad cow or spongiform encephalopathy for the cows and the cattle. And in the case of prions, you saw how some of that got into the food chain, and some individuals got the deterioration of their own brains. It's called Creutzfeldt-Jakob disease, but they usually put it with a V in front of it. About 140 people were sickened by it and eventually died because there is no treatment for this. Now, let me move forward. We'll get more into prions later on in the course. Most of these organisms are microscopic in size. They're often, in some cases, harmless. In some cases, they may be helpful. Okay, we have staph A and various forms of staph bacteria on the surface of the skin. Yes, even if you've washed and you did the shower and all that stuff. It is when they get inserted through a wound into the body that they will cause disease. Okay, and so that's what we want you to be aware of. Now, what is epidemiology? It's a branch of medicine that deals with the incidence, distribution, and possible control of diseases and other factors relating to health. One of the key tools for an epidemiologist are pattern recognition. In other words, if you see one person coming in with it and then you see another one coming into the emergency room, etc., and then you know that there's something related here. And this actually was part of what happened. Many of you are familiar with after 9/11, there were individuals, or was believed to be now just a individual, who had put dry anthrax powder into envelopes. Well, the problem with the envelopes was they go through the mail processing center. Sometimes you'd get some of this powdery stuff leaking out. And what happened was, I believe it was Virginia, the doctor in the emergency room was getting 1 and then 2 and then 4, etc., more cases of anthrax. And then she just picked up the phone and said, we got a problem here. And it's usually to the state health. And then it goes from there to CDC. Okay. Now, of course, epidemiology is a field that is focused on identifying patterns of morbidity. Morbidity is the rate of disease in a population. Mortality, this is another term for basically death, so incidence of death, and other risk factors. Etiology is the cause, set of causes, or manner of causation of a disease or a condition. All right? Now, if you get confused about this, think about this. If you go to, let's say, Alaska, and you're there for the very short period of time of a summer, the natives there, the people that visit Alaska will say, yeah, there's bazillion of these little, mosquitoes and insects and else, but you're not going to see the species that really will pass malaria. Go to the equator, and there is a high chance you will find mosquitoes that will be capable of transmitting malaria in their bite. Okay? And by the way, when I talk about mortality, I'm not just talking about death. I'm talking about a death rate due to a particular disease. Now, there are certain mathematical processes, and if I said math and you get all a tizzy, don't worry about this. They're very simple situations here, very simple calculations. When we talk about incidence proportion, this is the measure of the risk of developing the outcome of interest. What I mean by the outcome, either death or sickness. Usually it is calculated as the number of new cases in a given population at a given time period over the total population at risk for developing a disease during the same time period. Simple situation. Simple example, when we had the onset of AIDS, it was a very small note written in June of 1981 and the CDC has several publications, but one of them is very notable. It's called Mortality Morbidity Weekly Report, MMWR. And what they just tried to say was we have this strange cluster, small number of individuals in San Francisco within the gay community that is having these unusual set of symptoms, etc. Now, that was 81, okay? When you compare that to the total population of San Francisco, you wouldn't have as many, but then move it all the way to, let's say, 1991. And the number of new cases is going to be much higher. The exposed population during that period is going to be much, much greater. Okay? When we talk about the concept of prevalence, that's a measure of the burden of disease in a population. It's calculated by the number of existing cases over the population able to get the disease during a given time period. It may be represented as a rate or a percentage. So if I look at this, I'm going to say, oh, the number of current cases among the exposed population during the time period. Okay? So you might say, well, this is a disease that has tremendous prevalence because when we talk about the population that can be exposed, that has the potential of being exposed and hence sick, we can take, for example, Ebola. One of the problems with Ebola was you can have blood coming out from all the different orifices of somebody who is sick. They may also move around. They may have a seizure or something. And what you have is these bloody particles containing the virus floating through the air, and other people will get the particles into them, and then they are exposed and get sick. Okay? Now, we're going to talk about a couple of other different measurements. Mortality rate is the incidence of deaths in a population in a given time period. So the mortality rate is the number of deaths and over the total population in a given year. Nipah virus, which was seen in Eastern Asia. What happened was pigs would be the incubator for this particular virus. And they picked it up from basically the woodlands, etc. But when they passed it on to human beings, the mortality rate was 40%. In other words, total population over a given year, okay, number of deaths you had for all these people that were exposed, 4 out of, let's say, 10 would die. All right. Now, if we are interested in identifying leading causes of death, we might use the proportionate or mortality ratio, the PMR. PMR provides information on which causes of death might be the most important compared to others. Okay? So if you look at the formula there, you have number of deaths from a given cause in a given time period over total deaths in the same time period. This is kind of important because what you really have to do is, you may have heard of one of the conflicts that occurred during the period of COVID. You know, you'd have relatives and you'd have friends and you have this and that, and basically all the doctors were just marking off going, yep, COVID, COVID, COVID. even though it might have been heart attack, seizure, lung cancer, etc. And because they didn't know a lot in 2020, a lot of deaths were being attributed to COVID, and yet the total deaths, they didn't really winnow out all the other cases, those people that died from suicide, those people that died from cerebral hemorrhage, those people that died from a heart attack, those people that died from pancreatic cancer, etc. So this tool helps to kind of basically clean up the data so that you get a good handle on, is this something that could lead to a large number of deaths, or is it something that contributes to the total deaths, but it's not one of the major factors? Then we have case fatality rate. This is also known as the case fatality ratio. And it's basically a proportion of the total cases which are fatal. Go back to the point I was talking about, the Nipah virus. Okay, 40% deaths. That meant that 60% of those people got real sick, but did not die. Now, the reason they have the CFR is that it answers the question of how many people die of a disease out of a number of individuals who have been diagnosed with the disease in a given time period. Now, one interesting thing is, let's go over HIV for a few minutes. In the beginning, I told you that one of the first documented reports of deaths or of illness was in San Francisco. That was June of 1981. As you move throughout the entire period of the 1980s and going into 1990s, several things had happened. One of them was that a lot of people had been sexually active, but did not know they were spreading the disease to others. 2, the disease also was passed to others because of the fact that the disease was not neutralized in donated blood. So those that were either receiving whole blood or blood products such as hemophiliacs, etc., were starting to show up with it. And they'd be saying, no way, I haven't been sleeping with anybody or anything else. So you get a better handle of what is going on in the sense of how many may die compared to how many cases there are. Okay? All right. Now, let's take a look here for a second at this epidemiological principles. If you take a look on the y-axis, you have the crude mortality rate per 100,000. And if you look on the x-axis, you're going to see decade by decade from 1900 all the way out to 2020. And what happens is you notice that there are some serious changes that occur. Well, what's happening if we identify them versus non-identification of mortality? This one is without cervical cancer, the ID, and the non-ID is still there. If you notice the peak that takes off just before the 1920s, care to speculate what that is? That's due to World War I. And one other thing, what was called the Spanish flu, or it was called basically the Spanish influenza. Now, one of the things you got to keep in mind is that as we go from 1900s to 2020, principles of sanitation, hygiene, vaccination drove the burden of infectious diseases down. Now, not just that, but let's face it, overall medicine had improved. Antibiotics, and even in the hospitals, you had the personal protective garments, etc., so that persons were not transmitting a disease from one room to the other necessarily. Now, infections versus non-infectious disease, okay, and that's what the idea is here. The mortality for the United States since the 1900s. Scrap the bit about the cervical cancer if you want. Just do this from the perspective of infectious versus non-infectious disease. My correction. If you notice the annual mortality data for this figure, this is information from the Centers for Disease Control, and it also from the Prevention Wonder database. But what you look at is Spanish flu had a massive amount of deaths at that time, and then you continue down here. We had a death rate. We don't really understand everything about it. We have not totally identified it. But you had a lot of decrease here. Now, let's go into this a little bit further when it comes to infectious agents and things like that. When we have any infectious disease, it might be classified based on the causative agent. So it's important for us to understand classifying based on agent assists in identifying the treatment. Let me give you a historical point. If you remember a little bit ago, I talked with you about the Spanish flu. And guess what? They tried to actually make a vaccine for it. Now, mind you, this is 1917, 1918, 1919. But it never worked. And I'll tell you why. The late 1800s, about 1897, they had some evidence of a new microorganism they could not identify, though. So they called it a filterable organism. They called it basically virus. That's where it got its name. But if you can't see it, how do you know if you have basically developed the proper vaccine? This is further complicated by the fact that as they were doing this and making this vaccination, they didn't really know if what they were making was effective, but they distributed it. For historical perspective, it was 1935 when the electron microscope was developed. It was then that we could first see images of the first viruses. Now, electron microscope has a much more powerful magnification capability. We're talking 100,000, 200,000 versus the best light microscopes, which might be 1,000, in some unique cases 2,000 with the modern day technology. But we have certain limitations if we do not identify the treatment. For example, antibiotics work against bacteria. Antifungal drugs work against fungal infection. Most viruses are either prevented by vaccination or the symptoms are treated until the host can clear the infection. Now, when we talk about exposure, it's any factor that may be associated with the outcome of interest. What I mean by that is if you're exposed, and this particular factor is the one that you're really looking at, the outcome of interest would be obviously the infection. A couple of side notes here just to share with you. In the 1970s and the 1980s, people would come in with colds, etc. Now, colds are what? Caused by rhinoviruses. And they demanded the doctor give them antibiotics. Antibiotics were not going to work against a viral disease. Also, many antibiotics do not do anything for antifungals, for fungal diseases, I should say. It is specific antifungals which work on certain mechanisms or structures within the fungal organism that will lead to the clearing of the fungal infection. And not all antibiotics, I might add, work against bacteria. Some work better against gram-positive, some work better against gram-negative, etc. We're going to talk about this down the road because, unfortunately, we are ending up now with organisms, particularly bacteria, that are resistant to many of the different antibiotics. And even scarier are some of the diseases that really had been conquered or pressed down and reduced to minor instances, but now they're coming back full blast. And it's because they are resistant against not one, not two, but in some cases a whole host of different antibiotics. And you see that with gonorrhea, you see it with tuberculosis, you see it with Staphylococcus aureus. We call that MRSA, methicillin-resistant Staphylococcus aureus. Or we see this with some organisms that would normally be stopped by vancomycin, and they call it VRE, vancomycin-resistant enterococci, or etc. So let's see. Let's talk about the classification here. Now, if you had microbiology beforehand, this is going to be old hat. If you have not, don't panic. This is going to be sort of an all-encompassing things, and if you go through the videos, etc., you'll see a lot of the points and principles here. Okay, that we're going to basically be discussing. First off, bacteria. Small, single-celled organisms, they are called prokaryotes. They do not have a nucleus. Now, some of these organisms can be beneficial or detrimental. It depends on where they are in the body or on the body or got into the body. For example, you have Helicobacter pylori. Helicobacter pylori is one of these very unique bacteria that can withstand the low pH in the stomach. Okay? And yet they can contribute to stomach cancer, to stomach ulcers, etc. Then we have also Mycobacterium tuberculosis, and this is the causative agent of tuberculosis. And the scary thing is there are strains that are now out there that basically are resistant to some of the TB antibiotics that would normally work on them. Now, we go from bacteria to viruses. Viruses, of course, are the smallest pathogens. Usually they're encapsulated with, you have the DNA or the RNA, which contains the genetic instructions, and then they're encapsulated by what could be just a protein shell. We call those naked viruses, or the protein shell, but it is also then further encapsulated by sort of a lipid membrane and has certain extensions from it to allow for binding to the cells. Okay, those are called enveloped cells. A lot of these viruses, if you just put them on the table and you leave them there, they're not going to do anything. They don't make ATP. They can't replicate. They have to basically require a host, usually and this is to hijack the cellular machinery to make proteins, etc. So when the DNA or the RNA of that virus gets inside, it begins to hijack the biochemical functions of the cell. And this is all to replicate and make new viruses. And of course, examples can be HIV, they can be SARS, COVID, etc. Now, fungus, this is a little bit different. They represent their own kingdom. Okay, a few of the millions of fungi may infect the host. There's a bunch of fungi that doesn't do anything except maybe taste good on the top of your burger. But there are some, and they are very nasty. An example would be Candida auris. Now, you're going to hear this, yeast infections, etc. The yeast infections are something that would occur within particularly either the mouth or in the reproductive area for a female. And what happens is the environment becomes favorable for these yeast organisms to proliferate. Now, there's also coccidioidosis, and that is the causative agent of valley fever. Now, let me explain that for a minute. Valley fever, these are particles that make it up, basically long strings of the virus, of the fungi. What happens is the soil gets dry and they start breaking up into pieces and they go along the dust. And if they get inside you, they start to basically reproduce. And it can have amazing effects on basically the upper respiratory system, a little bit into the lower respiratory system, and into the nervous system. Okay? The Candida auris is the one that's going to be the scariest because at present, we are seeing this unique form of fungal infection that is resistant to every known antifungal drug. Now, there are different strains that have this, but like a lot of traits, microorganisms exchange some of this pieces of DNA and enhances the reproduction and the success of that newly recombined organism. Let's get to parasites. Now, parasites live in or on other organisms. Usually, like a virus, the parasites need hosts, but you got to understand something. The parasites may be much larger, so they can be seen by microscopes and things like that. And they do not require the host machinery to reproduce like viruses do, but instead they will lead to disease and in some cases death of the individual. And so we're going to talk about some parasites on the week that are basically more protozoan. Others that are going to be of the various types of helminths. In other words, like tapeworms, roundworms, flukes, etc. And the thing is, some of this can come from contaminated meat. Some of this can come from accidents where a fly has some of the eggs of the, let's say, tapeworm and lands on your food and flies away. And some of those eggs then get into you and eventually basically begin to hatch, grow, and individuals will have tapeworms in them. Now, prions. This took a long time for scientists, some of them, to accept it. And they in a lot of cases, gave Stanley Prusiner a lot of grief, but he was able to provide the sufficient research data and had very rigorous setups to demonstrate that this was a transmissible protein. Okay? No genetic information at all. And what it does, leads to abnormal folding of normal cellular proteins. So those that get abnormally folded become themselves prions. And this can lead to the deterioration of brain tissue, the spongiform encephalopathy that we talk about. The interesting thing is that it can be passed from species to species. And it can be found in humans. There is presently no treatment to cure it. Okay. Creutzfeldt-Jakob disease, that's more often you're going to see that with humans. Okay. Chronic wasting disease, that you see with mule deer and I believe elk in the West and in the Rockies. And yes, their brains are deteriorating. Okay. Kuru. Now, Kuru is an interesting one. I'll just move aside for a second. And this is what deals with a cultural issue. In New Guinea, the natives there, if somebody died, they would, at the ceremony, basically handle the person's brain. And in some cases, take a small piece of it and consume it. Okay? Well, what they were doing was passing on to the next generation Kuru, prion-based disease. As a matter of fact, when they stopped that traditional ceremony, that part of it, Kuru kind of stopped. Scrapie, I've mentioned to you before. You see it very commonly in sheep. Basically, since the British disaster, which caused the Great Britain, which had contaminated beef, it came from contaminated cows, which were fed sheep that had scrapie, so the protein products were fed there to the cow. Basically, they don't do that anymore. Now, by the way, you might say, well, why didn't it happen in the United States first? We have so many more cows. The way that we use our protein supplements, by the way, is by giving them extra grain in the sense of soy and corn and stuff like that, which bolsters their protein intake. Okay? And so we move from here to give you some perspective. Now, when I say emerging disease, a lot of people think, well, it just came out from the wilderness, That's true. Some of these do. And sometimes they will be eradicated and we won't see them anymore. Case in point, smallpox. But we also have to deal with the issue of reemergence. Now, reemerging infectious diseases, these are the diseases that once were major health problems globally or in a particular country. And then they decline dramatically, but are again becoming health problems for a significant proportion of the population. Case in point, malaria, tuberculosis are several examples. Now, just to help you also to think about this, smallpox. Smallpox has actually been eradicated globally. There are only samples of it that are held in 2 labs. One of them's at CDC, Center for Disease Control, and one is at the Russian facility called Vector. If you are, let's say, around 55, 60, somewhere around there, you may have gotten a smallpox vaccination. But after a certain point, the United States stopped vaccinating them, vaccinating kids and families and everything else. There is some vaccination going on in the military, because with so much of the global population vulnerable to smallpox, there was a point when smallpox was considered a potential biological weapon. Hey, if you release it, you could make lots and lots of people sick. And there was, the Russians had been very, very busy trying to make really nasty smallpox versions. One of them was called Aralesk, and smallpox can have up to about a 40% fatality. Aralesk is 100% fatality. So you get it, you die. End of discussion. And some of them were being engineered to resist against vaccines and other things too. Now, let's get to situations of how diseases can reemerge. For example, societal issues of war, displacement camps, migration, deforestation. One of the situations that happens is in some areas, in deforestation just alone, you have some of the flora and fauna that basically have been the normal reservoirs for some of these pathogens, but now they basically come into areas where they're humans, and so the disease can pass on to humans. Climate change. This may force populations to be exposed to pathogens. Sometimes when we talk about war, what we're really talking about is not just, on a battlefield, but disruption of sanitation, disruption of clean, clear water, hygiene, mutation of pathogens. We're going to talk a little bit about that when we talk more about influenza, okay? And how it changes a little bit, and thereby we have to get another vaccination of that particular strain of the virus for the next year. The other biggies include development of antibiotic resistance of the pathogen. This is serious because at present, as I've mentioned to you before, we have several different diseases that right now are resistant to a variety of antibiotics. We also have a fungal disease that is resistant to a variety of the antifungal drugs. And this pushes us back into how we either make new types of antibiotics, and there's going to be some hope about that. I'm going to mention that later on. Or basically, you're going to see a greater amount of death. Lack of clean water or sanitation. That can occur in a variety of situations, either because of a breakdown of a government, a breakdown of resources to maintain the water being clean, or the removal of trash, sanitation, etc. And oh yeah, by the way, sanitation means you also get rid of your trash. Take a look in New York City. Sometimes if the trash removal services are on strike, you get these huge mountains of trash as you're walking along the sidewalks. Well, what happens is not only is the trash there, but the rats that feed on them. And we're going to talk about that down the pike, because rats can be a very powerful vector for a variety of diseases, especially when you have a high population density such as New York City. You also have the situations of high populations in refugee facilities. And, of course, lack of public health education. These are all situations that can lead to a reemergence of an affecting disease. Now, there are a couple of tools that help epidemiologists to determine if a situation is occurring and how they can intervene. The first model here is the epidemiological triangle. It's a framework for disease transmission and intervention. You have three major elements. You have the agent causing the disease, the host, and the environment. Okay? So disease agents, whether they're bacteria, prion, viruses, parasites, and actually they've used this also for non-infectious situations of chemical or radiation agents. You have exposure within that environment, and you end up with consequences for the host. Now, each of the sides represents a link between agent and host and environment and agent. Okay? And these are very critical that you get down because you're going to see a lot of this down the pike. This tool is very important for public health because they also represent places for intervention. In other words, you break the links between any of the pairs on that triangle, and so the transmission cycle may also break down. Now, it's because of these linkages that many epidemiologists focus their attention on research, policy, and implementation. And one of the things that happens is if they can see, like I told you about Kuru, you had the prion, you had individuals with a certain situation where they would handle the person who had just died's brains or even take a piece of it. So you have this acceptable environment where it's culturally acceptable, and then you have the agent, and what you end up with is passing on the Kuru to the next generation. Okay? Now there's another, a little bit more sophisticated one. I favor this at times. It's called the disease pyramid. One of the interesting things is that this has been used extensively for studying plant disease outbreaks. But it can be equally applied in certain situations for human diseases. It is an advanced tool, and it takes into account time as a factor of disease onset. Now remember, sometimes you get exposed, and it might be some days or some week. Smallpox took anywhere from 7 to 21 days. Usually they want to say, the person that was exposed, it's going to take them 14 days. They kind of round it up. Okay? But you still have your pathogen, your host, and your environment. But the time, how long does it take to trigger the disease by the pathogen? Is there a time of latency of the pathogen within the host? This can happen. Think about it from another perspective. Many individuals had chickenpox. And this is before the onset of the chickenpox vaccine and the shingles vaccine. So they have chickenpox within maybe, let's say, a week. They've got a spouse or a brother or whatever that's got it also. The disease eventually dies down, but the virus hides in the trigeminal nerve ganglia. And then as the immune system starts to break down, when you start going into 50s, 60s, 70s, you can have an outbreak of shingles. And that is where the virus becomes active, but then works on only one side of the body and what they call one particular dermatome. In other words, it follows along the spinal nerve or the cranial nerve, and it's just painful, blistering, and it takes a while to recover. Nowadays, chickenpox is being heavily eradicated. I wouldn't say it's successful completely yet. And also, those who have had chickenpox we have gotten the as they get older, the shingles vaccine for protection. Some diseases may take only a few days before they show up, but here's the other trick, and you'll see this in chikungunya virus paper that I wrote. Within only about 7 days or so, the individual feels horrible. And then after that, the virus is gone, but the damage will continue for some time afterwards. And the damage is swollen, painful joints, etc. All right. As I said, the pyramid has been used for studying plant disease outbreaks. Now, that's except in the case where the fourth point is not time, but it's the microbiome of the plant, in which is also the host. It gets a little tricky. They have used also the disease pyramid to predict the effects of a biological weapon attack. Okay, so we've finished up here. What I encourage you to do is read the sections from both Netter's infectious diseases and the Orem text. So your work for this week: read the lecture, review the lecture PowerPoints, read all the supplemental articles in Module Week 1. View all the videos in Module Week 1. Participate and complete the discussion assignment for Week 1. Now remember, that's one that includes not you're just posting a original, but you're going to comment on another student's work. Okay. Now, some of the discussion assignments require you to review a particular week's videos or supplement articles. In this case, you're going to have a little bit related, but you will find from time to time that there will be sort of a bunch of these articles, and they're to help you to get familiarized and be able to effectively deal with the discussion topic for that week. And always complete the quiz for the week. And we'll see you soon.